Today's topic name is antibodies and types of antibodies. Antibodies is known as immunoglobulin. These are large Y-shaped blood proteins produced by plasma cells. They bind to foreign particles and invade them. These particles are foreign bodies that get attacked by antibodies. Uh, Antibody structures are Antibody has a Y-shaped structure made up of 4 polypeptide subunits. Each subunit has 2 identical light and heavy chains. The end terminus of each heavy chain forms an antigen binding domain with a light chain. These are 2 antigen bindings domains forming the arm of the Y shape. They are known as fragment antigen binding domains. The C-terminus of the heavy chains forms fragment crystallization domain, which helps in the interaction with the effector cells. All 4 polypeptide subunits are held together by disulfide and non-covalent bonds. The heavy chains of the antibodies contain a variable region and 3 constant regions. Each antibody has 2 identical antigen binding sites and they differ in the bodies. Types of antibodies. Antibodies are of 5 different isotypes. This classification is on the basis of their H chain. Let's look at the different types of immunoglobulins and their functions. First one is IgM. IgM is the first antibody produced in response to a microbial attack by B cell. It is the largest antibody and is found in a pentameric form. It circulates in the blood and lymph and constitutes 6% of the total antibody content in the serum. It is involved in agglutination and opsonization. It has a large number of antigen sites on its surface and therefore facilitates efficient activation of the immune system. Second one is IgG, the most abundant isotype in the plasma and comprises 80% of the total antibody content in the serum. It detoxifies substances that are harmful and recognize the antibody-antigen complex. It is transferred to the placenta through the fetus and protects the infant until its birth. Ig is divided into 4 subclasses: IgGL, IgG1, IgG2, IgG3 and IgG4. Among these, only Ig3 and Ig4 possesses the ability to cross the placenta. The third one is IgA, usually found in liquid such as breast milk, serum, saliva, fluids of the intestine. IgA is a breast milk protects an infant's gastrointestinal tract from the microbial activity. It constitutes 30% of the total antibody content in the serum and is divided into 2 subclasses, IgA1 and IgA2. Among these, IgA1 is highly found in secretion and it is also called the secretory immunoglobulin. Then fourth one is IgD. It is involved in the production of the antibody by B cell. It is present as a monomer and weighs around the 1,80,000 Daltons. It comprises less than 1% of the total antibody content in serum. It acts as a receptor on B cell surface and participates in B cell activation and differentiation. And then fifth one is IgE. IgE is present in a least amount around the 0.02% of the antibody content in the serum. These are present in the lining of the respiratory and intestinal tract. This is found in a monomer in the body and weigh about 2000 Daltons. The functions of the antibodies are blind to pathogens, activates the immune system, directly attack the viral pathogens, assist in phagocytes, they ingest cell by phagocytosis. Thank you.